Hello my dear students, this is Dr. Pooja, your original guru of biochemistry. Now in this present video, we are going to discuss the minor pathways in carbohydrate metabolism. Now whenever we talk about the minor pathways, basically now you must have understood what are the major pathways, what are the alternative pathways. Minor pathways means along with glucose, what are the other things which we are having in our diet and how they are metabolized in the body. So, because we know physiologically there are three main monosaccharides, glucose is there, galactose is there, fructose is there. Glucose, I told you, it is actually the hero in the body, right? But apart from glucose, we are having galactose, we are having fructose. And you will be surprised to know that fructose is considered to be the more sweeter sugar than glucose, or it is the sweetest. And in terms of absorption, if I say galactose is 100 times more easily absorbed than glucose, because galactose is a component of milk sugar. Right? So far we have discussed about the major pathways which include mainly glucose. We have studied the alternative pathways which again use glucose. Now we are going to study the minor pathways also about galactose and fructose. And it is so interesting that whether we are having galactose, whether we are having fructose, they are going to merge themselves into the pathways related to glucose. Okay. So first of all, let's start up with galactose metabolism. Now see, we all know that galactose, if we talk about in terms of absorption, again S-GLUT, sodium dependent glucose transporter is there which is going to absorb galactose also. Now galactose is again a monosaccharide, so it is highly soluble. So what would be the first step of galactose? The moment it enters inside the cell, it is phosphorylated. The moment we put a charge on it, it cannot go back into circulation. Again, it is going to disturb the osmolality. Though the first step is that this galactose will undergo phosphorylation. But here the main thing is that the enzyme here is galactokinase, because I have told you wherever we are writing kinase, that means it attaches inorganic phosphate. Or from where inorganic phosphate is coming? It is coming from ATP. And what would be the product? It would be galactose 1-phosphate. And this is the basic difference, because whenever we phosphorylate glucose, it always forms glucose 6-phosphate. But always remember, whenever galactose and fructose are phosphorylated, they form galactose or fructose 1-phosphate. So obviously their Km, their kinetic properties, everything would be different, right? Now this galactose 1-phosphate, one more thing which you people know that we have discussed in the chemistry of carbohydrates, that galactose is C4 epimer of glucose. And when I discussed about the chemistry, I told you the conversion of galactose to glucose takes place in the liver. And that is what exactly happens here. Now the reaction is this galactose 1-phosphate will combine with UDPG. Now what is this UDPG? It is again the activated glucose, which is uridine diphosphate glucose, which is also a precursor of glycogenesis, right? So there will be an interchange. So I can say that interchange means UDP galactose will be produced and glucose 1-phosphate will be produced. Simple interchange is there. And if we talk about the enzyme here, the enzyme will be galactose 1-phosphate uridyltransferase, because uridyl group is the one which is being transferred. Transferase. Now again, one very important thing I told you, whatever is produced in the body, it has to be utilized in the body itself. Nothing is wasted as such. So if we talk about glucose 1-phosphate, we know glucose 1-phosphate can be converted into glucose 6-phosphate. And simple, the name of the enzyme is phosphoglucomutase, and glucose 6-phosphate will form glucose and the body will get glucose. So I can say galactose can again be converted into this, and this UDP-galactose will be required in the synthesis of lactose, in the synthesis of, you can say, proteoglycan. So many are there. So again, nothing is wasted as such, and it is being reutilized again and again. Now on the other hand, what is happening is this galactose, obviously it is undergoing phosphorylation to form galactose 1-phosphate. On the other hand, this galactose will undergo reduction also. And the enzyme responsible for the reduction of galactose, because galactose is a aldohexose, so it will be aldole reductase. This is again a very, very important enzyme. Aldole reductase. And what is the name of the product? Because again I have told you in the chemistry that whenever any monosaccharide undergoes a reduction, it always forms the corresponding alcohol. So the sugar alcohol of galactose is none other than galactitol. And in some books, galactitol is given as dulcitol. So these are basically the synonyms. You can write as dulcitol also, you can write as galactitol also. So this is the summary of the metabolism of galactose. Simple: galactose undergoing phosphorylation forming galactose 1-phosphate. This galactose 1-phosphate combining with UDP glucose forming UDP-galactose and glucose 1-phosphate. UDP-galactose is required for the synthesis of lactose and other products, whereas glucose 1-phosphate will be directly converted into glucose. I am not writing the name of the enzymes as we have already discussed all the enzymes in detail. And galactose on the other hand will undergo reduction forming galactitol. Now the question is, what is the clinical correlation of the metabolism of galactose? So whenever we talk about the clinical correlation, we have to go at the level of the enzymes. So if you see, this enzyme can be absent, right? Or this enzyme can be absent, because both are different enzymes. So whether this enzyme is absent or this enzyme is absent, there will be increased concentration of galactose. There will be increased concentration of galactose 1-phosphate. Okay, now the clinical features will be different when this particular enzyme is absent, and the clinical features will be different when this enzyme is absent. So now we are going to study the clinical correlation of now see this enzyme galactose 1-phosphate uridyltransferase is also known as GALT, galactose simple transferase. And the most important thing which you have to remember here is, again I will go to the basics, the classification. This belongs to epimerase. An epimerase comes under isomerases, class of enzymes. Here it is GALT and here it is GALC, because most of the students get confused between GALC and GALT. K for kinase, T for transferase. Okay, so if GALT is absent or if GALC is absent, in general I can say there will be increased concentration of galactose, and when the concentration of galactose is more, it is known as galactosemia. So I can say that galactosemia will be due to increased concentration of galkinase, or you can say galtransferase. Now the question is, what is the difference in this galactosemia? What will be the difference in the clinical features when person is deficient of GALT enzyme or GALC enzyme? So first of all, we are going to take GALT, right? So first is GALT, or I will just write the full form just so that you people don't forget: galactose 1-phosphate uridyltransferase. Now see what will happen here. Simply we can say that there will be increased concentration of galactose, right? It will lead to increased concentration of galactose 1-phosphate here, as you can see here, right? Now, when there is increased concentration of galactose 1-phosphate, this enzyme is absent. Okay, so the concentration of galactose is continuously increasing. So I can say that first of all, there will be decreased concentration of glucose. Okay, now what will happen is, since this reaction is taking place in the liver, the increased concentration of galactose 1-phosphate is going to inhibit सबसे पहले glycogen phosphorylase. I'll tell you the reason. First of all, glycogen phosphorylase will be inhibited. The concentration of glucose will decrease and gluconeogenesis will also decrease. And these three major factors will lead to fasting hypoglycemia. Very, very important. Now, what is the biochemical reason behind this? That this process is actually known as sequestering of inorganic phosphate. Sequestering का मतलब क्या है कि this inorganic phosphate which is continuously being increased as galactose 1-phosphate is not free to use glycogen phosphorylase enzyme. मतलब, or you can say trapping of inorganic phosphate. Very, very important. So you can say that the phosphate, the inorganic phosphate, because we know glycogen phosphorylase, if you remember again, when we discussed about this is the rate limiting enzyme of glycogenolysis, I told you this is phosphorylatic splitting. From where that phosphate is coming, that phosphate is already there in the pool. But the pool which is giving rise to that inorganic phosphate is not now available. Okay, if it is not available, glycogen phosphorylase is not going to work. Glycogen breakdown will not be possible. Dusra, gluconeogenesis is also not taking place. On the contrary, glucose ka concentration is also decreasing. Okay, so all these factors will lead to fasting hypoglycemia. And then what happens is that the increased concentration of this galactose 1-phosphate is highly toxic to liver cells. It is highly toxic. Right. So what will happen is the child will undergo hypoglycemia. It is toxic to liver cells and it will lead to what? The main clinical features here will be, it will lead to vomitings. It will lead to diarrhea. Right. And it will sometimes, mimic jaundice because it is toxic. And sometimes there is a confusion between physiological jaundice and this jaundice. But there is a world of difference here because here there is increased concentration of this. And obviously phototherapy se we can, treat physiological jaundice, but this jaundice is due to the deficiency of galactose 1-phosphate uridyltransferase. And it might lead to flatulence also. So these are the main features which will be there due to the deficiency of this galactose 1-phosphate uridyltransferase. And the most important feature here is that since the concentration of galactose is more, it will be diverted to form more of galactitol because it will be metabolized somewhere else. And the increased concentration of galactitol is responsible for the formation of cataract. And this again we have explained in detail in the chemistry of carbohydrates that the cataracts so produced by the increased concentration of this galactitol is oil drop cataract, or we call it as bilateral cataract, which is different from the snowflakey cataract which is produced as a result of accumulation of sorbitol. Okay, so this is oil drop cataract, or we can call it as bilateral cataract. And this is known as classic galactosemia. So if the question is asked like this, what is classic galactosemia? You can simply say that classic galactosemia is due to the deficiency of galactose 1-phosphate uridyltransferase, or commonly we call it as GALT. And if we talk about the features, the main ones are it will lead to vomitings, diarrhea, jaundice, and the main thing is oil drop cataract is seen. And if we talk about the biochemistry behind that, simply you can say that there will be sequestering of inorganic phosphate, there will be trapment of inorganic phosphate, or inorganic phosphate will not be available for the other reactions. So obviously it will lead to defective use of glycogen phosphorylase, there won't be formation of glucose, the child will undergo fasting hypoglycemia. Then another feature, these are the clinical features. Then what are the biochemical investigations for this? So if we talk about the investigations, the first and the foremost one will be, if we talk about Benedict's test, what will happen? Benedict's test will be positive or negative. Since galactose is a reducing sugar, it has to be positive. Right. Now, Benedict's test being a reducing sugar, galactose will give. But if we talk about glucose oxidase test will be negative, which gives a clear hint that it has nothing to do with glucose metabolism. Okay. Now, we have to go for Benedict's test be positive hai, glucose oxidase test negative hai, then we go for mucic acid test. And mucic acid test is specific for galactosemia and it gives white precipitates. Very, very important. Now, mucic acid test is there, then others are enzyme based studies, means we have to carry out the assay of galactosemia, we have to check the activity of this enzyme. Okay. So these are the 4 very, very important points how we can investigate and we can conclude that it is a case of galactosemia. Now after this, then another important point we have to remember is what is the treatment. So the most important thing here which you have to remember is that breastfeeding is actually contraindicated in this. So I'll just write contraindication of breastfeeding. Kab tak? Till 4 years. So after 4 years, what happens is, after 4 years, there is one more enzyme which is lying there which will be activated and which will actually utilize this galactose. Okay. And the name of that enzyme is galactose 1-phosphate pyrophosphorylase. Pyrophosphorylase. So this enzyme actually activates galactose. So one can start using this because the enzyme which is actually required for metabolizing galactose is absent till 4 years. After 4 years, automatically this enzyme will start working. Very important. Right. So this is galactosemia and that too classic ones. Classic galactosemia is due to the deficiency of galactose 1-phosphate uridyltransferase. Now there is also one more enzyme into function. Okay. The enzyme is galactokinase. That's what I told you na. Either this enzyme can be deficient, either this enzyme can be deficient. Abhi we studied when galactose 1-phosphate uridyltransferase is absent. Now what will happen if galactokinase is absent? Now here you can clearly see that if this enzyme is absent, only one reaction is being affected. And what is that reaction? Only galactitol. It has nothing to do with galactose 1-phosphate because galactose 1-phosphate is another chapter now because wo kahan pe aa raha hai? It is being metabolized by this. So if we talk about deficiency of galactokinase, there is only one clinical feature associated, that there will be cataract formation. No vomiting, no diarrhea, no jaundice will be seen in galactokinase. And that is why it is known as non-classic galactosemia. Okay. So the second one is non-classic galactosemia, or you can say simply the deficient enzyme is GALC. or galactokinase. It leads to non-classic galactosemia. And we can simply say the main clinical feature will be cataract. Right? Why cataract formation will be there? Because it will be metabolized to form more and more of dulcitol or galactitol. So this is the metabolism of galactose, and what you have to remember is you have to just remember these two enzymes which are very, very important. Galactokinase, which converts galactose to galactose 1-phosphate, and you can say galactose 1-phosphate uridyltransferase, which is a epimerase enzyme which basically metabolizes galactose to form lactose, and ultimately it ends to form glucose. And these are the two metabolic errors associated with this. Then second, again the last one is fructose metabolism. Again, fructose is a very, very important, I would say, monosaccharide which plays a very important role again, because if we talk about the syrups and all, because fructose is more sweeter than glucose. Jitni bhi aerated drinks are there, या you can say wherever sweetness is required, this fructose is the one which is added. And fructose, if we compare it with glucose, this is considered to be the most lipogenic sugar also. Because again, if you go back to glycolysis, it is bypassing first step of glycolysis, because fructose ka kya hai ki fructose will directly, it will be converted into fructose 1-phosphate and metabolized, right? So what are the important points which we have to know about fructose? Ek to, it is the most lipogenic sugar. Now what do you mean by the term lipogenic? Genic means synthesis, lipo means fat. So you can say ki it can easily form या तो TGs or you can say VLDLs. मतलब it will give rise to fats immediately in comparison to glucose. Second important point is more sweeter. And if we talk about the transport, fructose is transported by GLUT5 and it is simply facilitative transport. And always remember, infusion of fructose will give energy in a more rapid and convenient way instead of glucose, because it bypasses the first step of glycolysis. This is very important again. So gives energy rapidly. Right? Now the question is again how fructose is metabolized. And yes, one more thing, it gives energy to the sperms also. Because if you remember GLUT5 transporters, where they take this fructose, okay, gives energy to the sperms. And I'll just write polyol pathway. Now, what is the metabolism of fructose? What happens is, since again it is soluble in water, it will undergo phosphorylation, right? So this fructose, when it undergoes phosphorylation, just like galactose, it is going to form fructose 1-phosphate instead of glucose 6-phosphate. This again you have to remember, because every enzyme is specific for its action. If we are phosphorylating glucose, the enzyme was hexokinase and glucokinase. And I've told you the difference. Glucokinase is present in the liver and this hexokinase is present in all the tissues. Whenever we talk about the metabolism of fructose and galactose, they are always phosphorylated at first position. And the enzyme required is fructokinase here and galactokinase there. Right? Again, utilization of ATP or ADP is there, and this is occurring again mainly in the liver. Okay. Now this fructose 1-phosphate, it has how many carbon atoms? It has 6 carbon atoms. So it splits up. And this is a very, very important enzyme in a sense that splitting enzyme is aldolase. And here you have to remember it is aldolase B. And since it has 6 carbon atoms, it is going to split up into 3 carbon atoms. And it is going to form DHAP and glyceraldehyde. Now you see how it will merge itself into glycolysis. Right? Dihydroxyacetone phosphate. Okay. And this glyceraldehyde, it will simply undergo kinase reaction and it is going to form 3-phosphoglyceraldehyde. And you can simply say that it will enter into either glycolysis or gluconeogenesis, whatever is the requirement of the body, or gluconeogenesis, whatever is there. Okay, so this is the metabolism of fructose. And one more thing, fructose is not going to undergo reduction. Why? Because if you talk about galactose and glucose both are aldohexoses, so they can easily undergo reduction. Fructose is ketohexose first of all. No doubt fructose will form sorbitol, but that will be via a polyol pathway. Okay, and it is not at all responsible for the formation of cataract. This is very important. Fructose metabolism mein you will see there is no formation of cataract. Okay, and this fructose does not have a threshold. Even if it has a threshold, it is very low. So what happens is, जैसे glucose has a renal threshold, galactose has a renal threshold, यहाँ पे there is no renal threshold. The moment concentration of fructose increases, it will be excreted in urine as such. Okay. So now see, if we talk about the enzymes again, this enzyme can be absent, this enzyme can be absent. Right. Now if we talk about aldolase B, the concentration of this will increase. If we talk about this, the concentration of this will increase. Now, if there is deficiency of aldolase B, what happens is it leads to hereditary fructose intolerance. So, absence of this enzyme leads to hereditary fructose intolerance. And absence of fructokinase will lead to fructosuria. Here we are not writing fructosemia. As in galactosemia, we are not writing galactosuria. Okay. So here you can say that absence of aldolase B will lead to hereditary fructose intolerance and absence of fructokinase will lead to fructosuria. Now, what are the main clinical features if we talk about, first of all, HFI. Now see, first is HFI or hereditary fructose intolerance. And here the interesting feature is the child, when he is born with this defect, initially he will be totally asymptomatic, no symptoms he is going to show. But yes, if he is given any fruit juice, anything which is rich in fructose, automatically it is going to aggravate the symptoms because the enzyme required to split fructose is absent. This is very important. Okay. So first of all, I'll just write the deficient enzyme. Deficient enzyme here is aldolase B. Now, one more thing I just want to tell you here, it cannot be aldolase A, because if you remember, aldolase A was the enzyme in glycolysis. Basically, aldolase is an enzyme which belongs to lyases. It has actually 3 main types. So, one is A, B and C. The basic difference is this A is present in all the tissues because it is an enzyme of glycolysis, but obvious B is present mainly in the liver and the kidneys. right? And C is present in the brain. To some extent, we can say B is also observed in intestines because GLUT5 is responsible for the transport of fructose there also, right? And if we talk about the substrates here, the substrate in aldolase A is fructose. If you remember again, it was fructose 1,6-bisphosphate. This was the substrate. And again in aldolase C, the substrate is the same, fructose 1,6-bisphosphate. And if you see the substrate here, the substrate is fructose 1-phosphate, right? This is the basic difference. And these are the type of aldolase. So whenever we talk about hereditary fructose intolerance, it is this, fructose 1-phosphate. Okay. Now, if we talk about again the clinical features, I can simply say that there will be increased concentration of fructose 1-phosphate. So, if fructose 1-phosphate is increased, there is what? Sequestering of inorganic phosphate here. Because this will not be converted into DHAP and 3-phosphoglycerolaldehyde. Okay, so I can say that since the concentration of this increases, again hypoglycemia will be there and same features will be there. Hypoglycemia will be there and this fructose 1-phosphate ka concentration, if it is increasing, again it will lead to similar to galactosemia. So, I am not writing the features again, galactosemia. But the most important point to remember here is no cataract formation. Very, very important, which is present in both classic and non-classic galactosemia. So, this is the main difference, right? And another important point here to remember is initially the child is asymptomatic, right? But when given fruit juice, always remember, but when given fruit juice, his condition deteriorates. Why his condition deteriorates? Because the fruit juice is rich in fructose. And the enzyme responsible for the degradation is absent. So, this is again a very important question which you have to remember. So, this is hereditary fructose intolerance, right? And second is what? Second is fructosuria. Now, in fructosuria, the deficient enzyme is what? It is fructokinase. Fructosuria, the deficient enzyme is fructokinase. Now, what happens here is fructosuria, there is excretion of fructose in urine. Why? Because fructose does not have renal threshold. So, it is going to increase and it will be excreted in urine. So, I can simply say that fructose has a very low or insignificant renal threshold, right? So, it will be excreted in urine. And since it is not hepatotoxic, so you can say that no other symptoms will be there and cataract is not there and fructose simply it will be excreted. So, obviously, if we have to, first of all, we will see the investigations, how we can say. So, investigations may again, same will be, if we talk about Benedict's test. Benedict's test will be positive. It has to be positive because it's a reducing sugar. So, I can say that Benedict's test is positive. Right? Then second test which we are going to carry is glucose oxidase test. Now, glucose oxidase test has to be negative here. Okay, then what are the positive tests which will be there? The most important one is एक तो rapid furfural test will be positive and Selvanov's test will be positive. Now, Selvanov's test is a very important test which is actually given only by ketohexoses, and we know fructose is a ketohexose, right? So, Selvanov's test will be positive. And then again we can go for enzyme based studies and all like that, but this will be the investigation. And if we talk about the treatment here, treatment should be simple. The diet should be restricted in sucrose. So, sucrose is restricted because sucrose is what it is. First of all, it is a disaccharide form of glucose and fructose, and if you remember in the chemistry also I discussed that sucrose we call it as an invert sugar also. Why we call it as an invert sugar? Because sucrose has both glucose and fructose. So, sucrose appears to be dextrorotatory, but due to more levorotatory power of fructose, it overweighs that. So, sucrose has more component of fructose, and that is why sucrose will always give Selvanov's test positive, because if we have to heat the sample automatically, sucrose ka bond, that glycosidic bond will be broken, fructose will be released, which will simply condense with the Selvanov's reagent to give a cherry red colored complex, right? So, this is fructose metabolism. Now, one thing which I have written in the starting only, that fructose is most lipogenic. So, what is happening here is that if you remember glycolysis mein, you know, glucose was the one, I would say glucose forms glucose 6-phosphate, then fructose 6-phosphate. Here what is happening is, and here you can say that ATP was utilized. Whenever fructose is being metabolized, fructose is actually bypassing this step. Okay, fructose directly it forms fructose 1-phosphate and then it can form fructose 6-phosphate. So, since this step is bypassed, so I can say fructose 6-phosphate can form easily acetyl coenzyme A, and acetyl coenzyme A can lead to the formation of TGs. And that is why it is most lipogenic sugar. This we will be studying in the metabolism of lipids, that how acetyl coenzyme A is forming triglycerides. Okay, as you can see that yahan pe this step is bypassed. And that is why we say if an infusion of fructose is given, it gives energy more rapidly than what glucose gives. Okay, and another important point to remember about this fructose is that fructose is required for the polyol pathway. So, I'll just give you a brief this thing. What is this polyol pathway? Because fructose is required to give energy to the sperms. So, basically what happens here is that this glucose, right, this glucose can form sorbitol and fructose can also form sorbitol. Right, again simply you can say that glucose dehydrogenase, fructose dehydrogenase, it forms sorbitol. Here we can say it is aldol reductase also, and it is, we can write simple dehydrogenase enzyme, right? And this sorbitol is required to give energy to the sperms. Or this can be either way also, glucose can be converted, because all are interrelated, I would say, like this. Glucose can be converted into fructose, fructose can be converted into sorbitol. Polyol means many OH groups, and many OH groups are present in all the alcohols, right? So, this is all about the minor pathways or all about the metabolism of galactose and fructose. So, I hope this topic is clear to all of you. Right, that's all in this video. Thank you so much.